Okay, we can start.
Good afternoon, everyone. I am Jashanpreet Kaur, Assistant Professor from the Department of Pharmaceutics, and I am delighted to serve as the coordinator for today's expert lecture session. Under the Golden Jubilee celebration of our institution, Amar Shahid Baba Ajit Singh Jujhar Singh Memorial College of Pharmacy has been organizing a series of expert talks to promote learning and research among students and faculty. Continuing this wonderful initiative, this afternoon session brings us another opportunity to gain valuable knowledge and inspiration. On behalf of Amar Shahid Baba Ajit Singh Jujhar Singh Memorial College of Pharmacy, I extend a very warm welcome to our distinguished resource person, Dr. Debapriya Garabadu, and to everyone present here. Before we proceed, I would like to share a brief introduction of our respected expert. Dr. Debapriya Garabadu is working as an assistant professor in the Department of Pharmacology, School of Health Sciences, Central University of Punjab, Bathinda. He obtained his MPharm and PhD degree from IIT Varanasi in the year 2008 and 2015, respectively. He is having teaching and research experience of more than 13 years in several reputed organizations like APJ Abdul Kalam Technical University, GLA University Mathura, and Guru Ghasidas Central University Bilaspur. More than 100 research review papers with the cumulative impact factor of more than 150 and an H index of 24 are added to his credit. He has served as a resource person in several national and international conferences. Several national level conferences and workshops are organized by him in association with the Indian Pharmacological Society, the Indian Academy of Neuroscience, and many more. Thank you so much, sir, for joining us today and for agreeing to share your insights on such an informative topic antiviral and immunomodulatory activities of Occamum basilicum in Japanese encephalitis. We are truly honored to have you with us. Sir, you may now please begin your session.
Thank you very much for inviting me. I would like to give my gratitude to Honorable Director Professor Silas sir and his team that he has invited me on this occasion. So may I start my presentation? Hello? Hello? Am I audible?
Yes, sir.
Hello?
Yes,
Yeah.
sir.
Okay. So may I start the presentation?
Yes, sir.
Hello? Okay.
Yes, sir. Please start.
Yes. The topic of the today's presentation is antiviral and immunomodulatory activities of Osimum basilicum in Japanese encephalitis. You know, I don't know the audience whether they are from B Farm or M Farm or PhD, but I would like to convince the audience that Osimum basilicum is one of the natural plant or the medicinal plant which is having a potential antiviral and immunomodulatory activity against this Japanese encephalitis virus, because this Japanese encephalitis virus causes Japanese encephalitis. So to introduce about this topic, I would like to um, bring these are the different outlines. So first, I would like to introduce Japanese encephalitis, which is a most common viral encephalitis in Asia as per the WHO guideline 2025. The initial case of Japanese encephalitis was introduced in the year 1871 in Japan, where more than 600 cases has been reported. As far as the etiology of Japanese encephalitis virus is concerned, it is isolated from the Japanese infected human brain in the year 1924, and the strain was Nakayama strain, which has been identified in the year 1935. This Japanese encephalitis virus is a mosquito-borne enveloped non-invasive RNA virus, which comes under flavivirus family. Now, as we can see from this figure, that this neglected tropical disease, which is Japanese encephalitis, is a global disease which has more than 1 lakh cases per year, and the death rate is about 13,000 to 20,000, and it is mostly prevalent in the 24 countries, including South and Southeast Asian and Western Pacific regions. As per the WHO guideline 2025, it has been reported that that India has most number of cases of Japanese encephalitis. Subsequently, China, then Philippines, Vietnam, and Nepal, and recently Australia and Benin also reported Japanese encephalitis. You can see from this figure that how this Japanese encephalitis travels from one host to another host. The carrier or the vector is the mosquito, that is Culex species, and it is having two different hosts, that is amplifying host and dead end host. Now, what is the difference between amplifying host and dead end host is that in amplifying host, the virus will multiply, but it cannot produce any symptoms or it cannot cause any damage to the amplifying host. But in the contrast, you can see that the dead end host are humans, horse, and other mammals. What they do, that the host, that virus multiplies in that human and horse, and subsequently you will find several deleterious symptoms which can lead to the death of that host. So this is the major difference between amplifying host and dead end host. And this mosquito transfers this virus from amplifying host to dead end host. So this is about the virus cycle. Now you can see the different phases of the virus, which once it enters into a human being, then the first phase is the incubation period, where it lasts for 5 to 15 days, and in that phase you will not find any symptoms. Then subsequently, prodromal phase will come, where you will find several non-specific symptoms like diarrhea, headache, high fever, right? Vomiting. All these cases will be observed for 3 to 4 days. Then you will find encephalitis. Once it will still proceed, there will be a acute phase of Japanese encephalitis, where the patient will experience confusion, paralysis, abnormal posturing, Parkinsonian symptoms like movement disorders, seizures, even coma. But once the virus will get eliminated from the patient, or the encephalitis will be recovered, it is almost 20 to 30% cases are severely affected. So in that case, the mortality rate lasts for 30%. But in other cases, the patient recovers from this encephalitis. And once the virus exists in the human physiological system, there will be several neurological complications. So mostly we are looking for those complications, but which will be experienced because of this virus infection. So therefore, we need to have a antiviral drug against this Japanese encephalitis virus, which could be effective to minimize these neurological complications induced by this Japanese encephalitis virus. Now you can see from this figure that the management of Japanese encephalitis, the most prevalent mechanism or the therapies, that is the supportive therapy during this infection, that is antipyretics and IV fluids and vanitors. They are given as a supportive measures in this Japanese encephalitis. However, vaccine is one of the best choice in this condition, because these Japanese encephalitis have 5 different genotypes. Now I would like to address what are the different type of genotypes which are available in Japanese encephalitis virus. So there are mostly five genotypes which are available in Japanese encephalitis virus. That is 1, 2, 3, 4, and 5. In India, Japanese genotype 5 is mostly prevalent, and whatever the vaccines has been developed, that is according to the genotype 5. Now, what happens? The lack of long-term effectiveness of the vaccines, lack of cross protection, and a re-emergence of genotype 5, and a low vaccination coverage and malnutrition, they all decrease the popularity of a vaccine among Japanese encephalitis patients. So that is one of the reason which is responsible to consider a potential antiviral drug against this viral infection. Therefore, what are the other drawbacks of this measurement is that vector control. So vector control mostly is done in those areas where this pig and rice farmings are being done, and there are certain factors which are leading to the limitation of this vector control. So therefore, we can consider for the antiviral drug against this virus. But several molecules have been evaluated in preclinical and clinical condition. However, there is no potential drug against this Japanese encephalitis virus is available in the market. So therefore, it is our prime interest to develop or to establish a potential therapeutic drug against Japanese encephalitis viral infection. Now, after several literature review, what we could find is that Osimum basilicum is one of the potential medicinal plant which is effective against several antiviral drugs, viral infections. However, it has been reported that the active constituents of Osimum basilicum and the extracts of Osimum basilicum and the essential oil of Osimum basilicum has been reported for antiviral activity against different viral pathogens, including aviviral family. However, the effect against Japanese encephalitis virus has not been evaluated till date. At the same time, this plant has been reported for antioxidant, anti-inflammatory, immunomodulatory, and organoprotective activities against several neurological disorders. Therefore, as this virus is prevalent or it is highly available in the brain, and that is the reason why it causes encephalitis. So the rationality of the study is that plant has a significant bioavailability in the brain, and it is having potential antiviral activity against other viral diseases, including the family of this virus, that is the flavivirus. So therefore, we have considered this plant as a potential therapeutic solution against Japanese encephalitis infection. Now, you can see from this slide that despite the ongoing preclinical and clinical studies, there is no clinically approved anti-JV drug. Thus, there is a need to develop an antiviral drug against this viral infection. Several phytoconstituents of Osimum basilicum has been reported against antiviral and viral pathogens, and there is yet to be established this Japanese encephalitis experimental model. So these are the research gaps, and therefore the hypothesis of the study is that we have considered potential four different targets of Japanese encephalitis. That they are envelope protein E, then NH3 helicase, NH3 protease, NH5 RDRP, that is RNA dependent RNA polymerase. As you can see from this, from the biochemistry point of view, that envelope E protein of the virus is responsible for the attachment of the virus with the host protein at the cell surface, so that the genetic material of the virus will enter into the cell, and then it will uh, multiply and it will form the virus, which could be infectious to cause other cell infection. Now, in the when the genetic material of the virus enters into the cell, the RNA gets multiplied. That is called the RNA replication, and at the same time, RNA can form the proteins which are responsible or which could be effective in the formation of the infectious virus particle. So, NS3 protease is one of the non-structural protein which is responsible in the protein synthesis of viral protein synthesis. But at the same time, NS3 helicase and NS5-RDRP, they are responsible for the RNA replication. So because the RNA replication and protein synthesis are majorly responsible for the synthesis of the viral particles or the infectious virus, we have targeted these three different targets. That is NS3 helicase, NS3 protease, and NS5-RDRP, along with the envelope E protein. Now, what is our objective was that first we need to screen the drugs, and what we have considered that from the database we have selected around 265 compounds from the Osimum basilicum plant, and those were docked with these four different targets, and then we have gone for the potential molecular docking, then molecular dynamic study, and then we have selected the drug which could be considered for in vitro and subsequently in vivo study. So the aim of the study was to screen and evaluate the antiviral activities of phytocompounds from Osimum basilicum in Japanese encephalitis experimental models. Now the first objective was to screen phytocompounds of Osimum basilicum against four drug targets of JV using an in silico model. Subsequently, uh, the second objective was to evaluate the antiviral activity of the selected compounds from the objective 1 in JV challenged cell line study. Then objective 3 was to evaluate the antiviral activity of the selected phytocompound in JV challenged mice. Then to evaluate the neuroprotective effect of the selected compounds in JV challenged mice. So in the first objective, you can see that 265 phytocompounds of Osimum basilicum along with their reference compounds were selected for the in silico study where we have used this Kroninger software. Then subsequently, the molecular dynamic study where RMSD and RMSF, other molecular dynamic parameters were considered, and we have selected a few compounds for the in vitro and then for in vivo study. You can see that after docking, we have selected three compounds, that is chichoric acid, rutin, and salvinoleic acid, which were having high binding score. From the two-dimensional structure, you can see that these compounds are having good interaction with this envelope E protein, NS3 helicase, NS3 protease, and NS5-RTR. Here we have summarized their uh, docking scores. Okay. For envelope E protein, we have considered mycophenolate as a reference compound. For NS5-RDRP, we have considered GTP as a reference compound. For NS3 helicase, we have taken ribavirin as a reference compound. And NS3 protease, we have considered paritaprevir as one of the reference compound. And you can see the chicotic acid, rutin, salvinoleic acid along with the reference compound are the docking score. And you can see that the better the docking score, it indicates the stronger binding interaction and the better the efficacy of the hit molecule against the particular disease condition. As you can see from the intermolecular contact analysis, you can see the chicoic acid has a hydrogen bonding with the alanine 35, lysine 336, and a lysine 336. And these blue colored amino acids are similar with the reference compound. So this indicates that these are the amino acids which are responsible for the interaction between the drug target and the molecule. Similar observations you can see from the NS5 RDRP, that is RNA dependent RNA polymerase. The reference compound has the interaction, and the amino acids which are being involved in the interaction between the reference compound and the drug molecule, the drug target. The similar amino acids you can see in the chicoic acid, rutin, and sulfonolic acid. So more amino acids are being involved in a compound. That means it is more strength. I mean the stable interaction. Right? In NS3 helicase also you can find chicoic acid and rutin has the common amino acids with the standard, but there is no common interaction between sulfonolic acid and the standard compound, that is ribavirin. So that is the reason why we have not included sulfonolic acid in this data set. As you can see from the NS3 protease, the chicoic acid, rutin, and sulfonolic acid has the same common amino acids which are being involved in the reference compound. So after doing this molecular docking study, what we can observe that there are certain limitations of molecular docking. So there, because lack of comprehensive information on protein flexibility, absence of solvation and ion effects, and inadequate scoring function and unreliable estimations of molecular affinity, it is necessary to validate the docking results through this molecular dynamics simulation study. So therefore, we have further studied molecular dynamics. So in the molecular dynamics study, we have considered 5 different parameters, that is RMSD, R-GIRI, SASA, PCA, and protein ligand contact analysis to observe the stability of the protein ligand interaction. Now in the RMSD analysis, what we can see that there is a large number of fluctuations, and the line what has been observed with the standard compound. You can see that this mycophenolate in C alpha and ketone protein, there are two different uh, figures. Now, in the C alpha means it is the backbone of this uh, drug target or the protein, which is highly stable. So, in that case, you can see that the interaction with this chicoic acid and rutin shows more or less similar activity along with the reference compound. And these observations you can see in both C alpha and feton protein analysis. And therefore, from this protein ligand RMSD analysis, it can be observed that chicoic acid and rutin are good candidates, um, that they are providing stable interaction with this envelope E protein. Similar observations you can find in this NS3 helicase, NS5 RDRP, and NS3 protease analysis. Now, after this study, again we have considered the protein ligand contact analysis. Now, what are the when there is an interaction between the drug molecule and the particular amino acid of the drug target, what kind of interaction it is, whether it is an hydrogen bonding, whether it is an uh, you know um, pi bonding or hydrophobic interaction. All those interactions we have summarized here, and these are the the red colored amino acids which are being involved. in the interaction, and they are giving the highest number of interaction fraction. So this indicates that these amino acids are being involved in stabilizing the drug and um, protein interaction. Similar observations you can find in NS5-RDRP, NS3 helicase, and NS3 protease. Now, after this thing, again we have considered this principal component analysis. Now, the principal component analysis gives us the strength about the binding interaction, the interaction between the drug molecule and the drug target. So from this study, you can see this highlighted blue color indicates that in the PC1, that is the principal component 1 and principal component 2 analysis, and the principal component 1 are depicted in red color and principal component 2 is depicted in black color, and this distribution indicates that the strength of the drug and protein interaction. So here also we have found that chicoic acid and rutin, they provide good stability with the drug target, that is NS3 protease, NS3 helicase, NS5 RDRP, and envelope E protein. So as a summary, what we can observe from this study is that in molecular docking analysis, chicoic acid and rutin showed better docking score with all drug targets of JEB except with NS5-RDRP as compared to their respective standards. Hydrogen bond, salt bridge, and pi cation interaction were observed in this ligand and protein interaction. As far as the MDS, that is molecular dynamic simulation analysis, we observed that chicoic acid and rutin demonstrated better stability from these parameters. So thus we have considered chicoic acid and rutin for the in vitro evaluation. Now in the second objective, we have performed the antiviral activity of chicoic acid and rutin in cell lines. So the cell line what we have considered, that is Vero cell line and SHSY 5 cell line. And the JV strain what we have considered, that is P20778 strain. And this virus was procured from RCB, that is Regional Center for Biotechnology, Faridabad. And the parameters what we have considered for the cell viability and cytoprotective assay, right? For plaque analysis, we have considered time of addition assay and virus entry inhibition assay, then immunocytochemistry, doses, H2DCF, DA for ROS level estimation. So in this study, the cell line were uh, um, considered in this cultured, and the drug treatments are different phases, that is pre, co, and co-treatment and inactivation, like that 5 different stages we have taken, and we have inoculated the drug to the viral infected cell cultures. So these are the different the parameters that we have considered for dosage study, then immunocytochemistry, and H2DCF analysis. Now we can see from the figure that there is a different cytotoxicity profile of chicoic acid and rutin. So from this figure, you can see that cytotoxicity or CC50, that is cytotoxic concentration of routine, chicoic acid is about 16 micromolar and 91 micromolar for chicoic acid, and the CC50 for routine, it is more than 2000 micromolar. So it indicates that the routine is more safer than chicoic acid. However, we have considered both these drugs, chicoic acid and routine, in SHSY5 cell line studies for the antiviral activity. Now, chicoric acid routine demonstrated significant cytoprotective activity in pre and post-treatment approach. So here we have taken pre-treatment, post-treatment, and post-treatment analysis. Pre-treatment means before the viral infection, we have given the drug. Post-treatment means we have given drug along with the virus simultaneously, and in post-treatment means first we have injected virus or introduced virus to the cell culture, then we have given the drug, observe the antiviral activity. So in that way, three different models we have taken in the cell culture study. But what we have observed is that chicory acid and routine, they exhibited significant cytoprotection in pre and post treatment approach. You can see the different phases. Again, we have considered that is pre-treatment, co-treatment, continuous treatment, virus inactivation, and attachment phase, where we have considered the IC50. The IC50 range for chicory acid, it was observed that it is about 11.03 to 24.04 micromolar, and for routine it is 16.45 to 26.84. And in this different the objective of considering different models of this study is that to address the viral entry, replication, and the virucidal activity. From the immunocytochemistry point of view, what we have observed is that this chicory acid and routine show dose-dependent antiviral activity in JP challenged cells. From this figure, you can see that the envelope E protein in the different, you can see from the JV, you can see this DAPI means the number of cells are there, and E protein, it is highly prevalent, and merged case, you can see this JV, the envelope E protein is highly uh, observed. But in case of the treatments, the chicory acid 8 micromolar and chicory acid 16 micromolar, and even in the routines, the number of envelope E protein are lesser. So this indicates that the viral loads are being decreased with the treatment condition. And even we have considered this Western blot analysis where we have taken NS5 protein. And you can see from this figure that after the treatment with chicory acid and a routine, significantly this NS5 protein is being decreased compared to the disease group. From, from the flow cytometry analysis, what you can see is that the apoptotic cells are being increased in this viral infection condition, and with the treatment of chicory acid and routine in all these doses, there is a decrease in the number of apoptotic cells. This indicates that the cells are being recovered, or it is being protected with the presence of the drug. And similar observation you can see from ROS, generation of ROS. So with the JEV infection, the extent of reactive oxygen species generation is high, and as compared to the this JV infection, the treatments are being decreased with this ROS uh, concentration. So as a summary, that this chicory acid and routine both are having good antiviral activity against this viral infection. However, uh, routine is more safer than chicory acid. That is the reason why we have considered routine for the further antiviral activity in the animal model. So in the objective 3 and 4, we have considered the animal model where we have considered the C57BL6W wild type mice of either sex, and the JV infection was monitored with the help of this clinical sign scoring and survival analysis. Further, the plaque assay, immunoblotting, or PCR and other biochemical assay and immunohistochemistry are being considered to evaluate their potential antiviral activity in this animal model. So in this condition, we have considered three groups, not three groups, it is only five groups. That is the control group, JV group, and the treatment group have three different doses, that 10, 25, and 50 micromolar per hour. So in this study, we have taken the lethal model where as 10 to the power 7 um, plaque forming unit per ml has been injected into the animals, and it can be observed that within 3 to 4 days the animals will experience death. And then we have considered the treatment, and we would like to know that after this 15 days what will be the condition. So we have observed this study for 15 days. These are all the parameters what we have considered after this 15 days of study. We have isolated the brain and then homogenized, and brain lysate we have taken, and that has been considered for plaque assay, immunoblotting, RT-PCR, biochemical analysis. So these are the methods what we have considered to isolate the brain sample. Now you can see routine delays the JV infection, JV induced morbidity and increase the survivability of mice dose dependently. From this figure, you can see how this survival rate is being increased. You can see the routine 50 mg, it is around 40% of the animals were survived. Hmm. Routine also exhibited significant antiviral activity by suppressing infectious virus load, then viral RNA load, then viral protein expression, but not immunomodulation, that is the interferon and ISGs. So you can see from this figure is that this 10 mg per kg body weight did not significantly reduce the plaque forming units. However, 25 and 50 dose, they are significantly decreasing the plaque forming unit. This indicates that those infected animals, this viral load is decreasing significantly. Now, similarly, if the viral load is decreasing, it doesn't mean that the number of RNA will be decreased. To further elucidate that thing, we have also considered this RT-PCR study where we have estimated the number of viral RNA. So what we can observe from this thing is that you can see the 25 and 50, they are significantly decreasing the number of viral load, and that is viral RNA. In the same time, NS3, NS5 protein expressions were significantly decreasing. So all this indicates that this routine at the dose of 25 and 50 was significantly effective antiviral agent in JV infection. Similar observation also being reported while considering this NS3 protein expression in immunocytochemistry and EBA1, that is the macrophage activation, right? While considering the neuroinflammation, we have considered TNF alpha, IL-6, pro-inflammatory genes, that is NLRP3, ASC, IL-1 beta, then 18, IL-18, then caspase-1, and all these inflammatory markers are being decreased while treatment with routine 25 and 50 mg per kg body weight in both brain and serum samples. And similar observation is also being reported while considering the oxidative stress, that is the extent of ROS formation. So as a summary, routine 25 and 50 kg body weight showed significant antiviral activity by attenuating infectious viral particles, viral RNA, and viral protein expression in the mice brain. In also, the routine 50 mg significantly increased the survival rate and attenuated the encephalitis symptoms in the JV infected mice. Routine significantly mitigated JV induced neuroinflammation by decreasing microglia activation, inflammasome markers, and pro-inflammatory cytokines and ROS level. So these are the references. I would like to address my collaborators, that is Professor Arup Banerjee from Laboratory of Virology, RCB, Faridabad, Professor Suresh Tarija from Department of Pharmaceutical Science and Natural Products, Central University of Punjab, Batinda, Dr. Pramod Kumar Kuswa, Department of Microbiology, Dr. Jyoti Prakash from Department of Zoology, Central University of Punjab, Batinda. These are my groups who have been work for this study. I would also like to uh, you know, uh, give my gratitude to our Honorable Vice Chancellor, Professor R.P. Tiwari, for providing us the necessary facilities and giving motivation to work for the society. And these are my labs, and these are some pictures of Central University of Punjab. Thank you so much.
Thank you so much, sir, for such an insightful and engaging session. Your talk on Osmium basilicum has truly enriched our knowledge and inspired us towards future research. We are grateful for your valuable time and contribution. Again, thank you so much, sir.
If there is, if someone wants to ask any question, I'm ready to answer. Hello. Hello.
No, sir. No any question.
Hello.
Hello.
Yes, yes.
No doubt, sir.
Okay. Okay then. Thank you. Thank you so much for
Thank you, sir.
inviting me. Okay.
Thank you, sir.